Welcome everyone to today's event, Microbes as Therapeutics, presented by SGInnovate and Singapore Consortium for Synthetic Biology, or SINERGY. I am Ziying from SGInnovate, and if you're new to us, we are a government-owned organization in Singapore with the mission to build deep tech innovations from Singapore for the world. We invest in and help build deep tech startups in the fields of artificial intelligence, medtech, and quantum tech, just to name a few. We work with entrepreneurial scientists and clinicians to help bring their innovative research from lab to market, and we also develop tech talent and engage with a vibrant deep tech ecosystem. Today, we are glad to have Dr. Jeremy Lim, Dr. Yan Tan, and Dr. Jonathan Kotula present on the latest developments in translating microbiome research into therapeutics applications. This will be followed by an exciting panel discussion moderated by Dr. In Young Huang. Just some house rules. During the webinar, if you have any questions, please post them in the Q&A box at the bottom of your screen. And do feel free to use the chat box to connect with our wider community. Without further ado, let me hand over the time to Ning Mao to say a few words. Ning, please.
Thank you, Ziying. Hi, everyone. My name is Ning. I'm currently an associate at UBX Ventures. Today, I'm very glad to be co-hosting this webinar with SGInnovate. Let me first introduce the SINERGY and the speakers today. So SINERGY is the Singapore Consortium for Synthetic Biology. It is funded by the National Research Foundation of Singapore. It currently represents 5 research institutes and 17 industry members, and is also growing its network. SINERGY is committed to promote academic industry collaborations and technology translation through organizing events and providing research seed grants for synthetic biology projects. Companies who are interested in synthetic biology research are very welcome to join the consortium. Today's webinar will be the third one in the series on synthetic biology and entrepreneurship. Again, we have a very exciting lineup of talks and discussions. First, we will hear from Associate Professor Jeremy Lim. Jeremy is the CEO and co-founder of Asian Microbiome Library, or AMiLI. It's Singapore's first precision gut microbiome company. As a medical doctor trained in surgery and public health, Jeremy is also a long-term advisor to a number of businesses and institutes, and he's also the director of Saw Swee Hock School of Public Health at the National University of Singapore. Next, we will hear from Dr. Yan Tan, who is the CEO and founder of Xbiome Biotech, an AI-driven microbiome drug development company based in Shenzhen, China. Dr. Tan has a strong computational biology and bioinformatics background with research experiences in the Broad Institute, and he obtained his PhD at Boston University. He's also named as one of the 35 under 35 innovators in China by the MIT Technology Review this year. Last but not least, we have Dr. Jonathan Kotula, who is the head of science at Felix Biotechnology based in California. At Felix, he leads the R&D team to engineer phage to treat antibiotic-resistant infections. John got his PhD in biochemistry and molecular biology from Duke University, and also did research work at Harvard Medical School and the Beast Institute. Knowing John for a few years now, he's always been at the forefront of engineering microbiomes for therapeutics development, with experiences at SYNLOGIC and Caribou Biosciences. So I'm very excited for all 3 speakers. They will offer their very different perspective on how microbiome research can translate to bring clinical benefits. After their talks, the speakers will join a panel discussion with Dr. In Young Huang. Dr. Huang is a research assistant professor at National University of Singapore with rich research experiences in synthetic biology and microbial therapeutics development. After this discussion, we have about 10 minutes for Q&A, so we encourage the audience to type their questions during the talks and discussion. So without further ado, we welcome the first speaker. Jeremy, please.
All right. Thank you, Ning, and a very good morning and to everyone, or rather to most of you, and a good evening to to really John. If I can share my slides very quickly, and while it's loading, I'll just introduce myself. And I will state up front, I'm not a synthetic biology person. I am a medical doctor, but my training was not in the gut microbiome. I focused on prostate cancer when I did my training in the US. And I must confess that when I was in medical school, all that we learned was that the microbiome was not very useful, and that you didn't need to study too much. There's probably all of 2 lines in any medical textbook about the microbiome, with some implications for vitamin K metabolism, but that was about it. So how much has changed in the last couple of years? And I entitled my well short thoughts here, "Wanted Premium Poop," because I wanted to acknowledge that this is the headline of a Straits Times article about AMiLI a couple of months ago. And I must say that the Straits Times headline writers are so incredibly creative, but that's really exactly what we are about in one of the value levers that we are carrying out. I don't need to preach to the choir. Really, microbiome science, everyone is talking about it. You know that when gut microbiome science makes the jump from obscure journals into mainstream journals like Nature and Science, and then into BBC, CNN, and Forbes, you know that it's really for prime time. And no less than Bill Gates has articulated that he believes that microbiome science is the way to solve world hunger. So what then are we doing? And AMiLI is really a compression of 3 words: Asian Microbiome Library. And what we are is fundamentally an effort to do 3 things. 1, we want to build an Asian-focused multi-ethnic gut microbiome reference library that really takes the different ethnic groups across Asia, pairs it with a lot of metadata around health data, biochemistry, wearable data, and put this together and make it very powerful as a platform for researchers, for industry to have that discovery opportunity. Because as we look at microbiome science, we are really at the beginning of what is a very exciting, and we certainly believe to be a very impactful journey. Earlier this month, we also launched Singapore's first gut microbiome sequencing service called Biotechnology. Right, and this is an effort for us to really create the awareness about gut microbiome science, bring the public on really on board, and create the milieu for food manufacturers, for biopharmaceutical companies to know that there are markets that good gut microbiome science can really impact really persons' lives. A little background about us, as I had alluded to earlier, that I'm a medical doctor, but I'm not a gut microbiome scientist. Really, as CEO of the company, my job is to enable good science. And these guys behind, my co-founders, Dr. David Ong is the head of gastroenterology in the National University Hospital. He did the first gut microbiome transplants in this part of the world back in 2013-14. Jonathan is Singapore's first clinician scientist focused on the gut microbiome, did his 2-year fellowship at the Broad Institute, just like Dr. Yen Tan, who will be presenting after me. Right, and this is Dr. Chris Ting, who is a laboratory medicine specialist. So you can see that I'm the least useful guy out of the 4 of us, and really my role is to enable the resources, forge the meaningful partnerships for us to acquire data, for us to have those industry and really research collaborations to enable good science to essentially propagate and bring good science into Asia. One of our discomforts with the state of gut microbiome science was really that all the exciting things largely were happening outside Southeast Asia. The North Americans, the Europeans, and increasingly the Chinese, the Japanese, the South Koreans were all doing great science. But in our part of the world, Southeast Asia, the number of clinical studies could literally be counted on just 2 hands amongst the over 1,000 clinical trials that were ongoing. So we then decided to change that. We decided that in addition to doing our own research, we would do 2 things. One, we would set up the Asia-focused multi-ethnic really database I had alluded to. And secondly, we would also put in the energy to establish Southeast Asia's first and only FMT bank. And the Straits Times, I want to acknowledge, have been very, very supportive, putting out a number of different articles to help us to publicize. And hand on heart, I've been so impressed by Singaporeans who have stepped forward to become donors, telling us that they believe in the science, they believe in this mission that we have to save lives and to impact human health. So I just want to share very quickly that this is a very inclusive platform. Beyond us as the co-founders, we have also really brought on board luminaries from Australia, from Malaysia, and really so on. And I cannot overemphasize that this is not about AMiLI. This is about gut microbiome science and bringing the science into clinical, into public health applications to benefit many more people, right? I know that my two co-speakers are going to speak about computational biology and synthetic biology. And as the poor clinician, let me just focus the rest of my time on essentially FMT, or which is short form for fecal microbiota transplant, which is basically it is a poop transplant. What is done is that stool is taken from healthy donors, and you will not believe how hard it is to find truly healthy donors. For the last 1.5 years, I've been spending virtually every Saturday morning screening individuals who come into our clinic believing that they are healthy. But out of 100 people that I see, ultimately only 2 will be eligible to be microbiome donors or to have their stool put into the stool bank ready for transplants. And the state of the science for many years had been that stool transplants were the standard of care for an infection called Clostridium difficile. But increasingly, we now know, as really Hippocrates said thousands of years ago, that all disease begins in the gut. And today, beyond Clostridium difficile, we are starting to see applications in inflammatory bowel disease, in irritable bowel syndrome, and in many more. What I wanted to just end off is by saying that many of these studies are not just confined to mice models or anything like that, but the future is being discovered in real time. There have been many human studies, relatively small, but extremely promising. And I just wanted to highlight 3 of these studies that came out in the last 12 months in what we consider to be the 3 most exciting areas. One is in the immune system modulation. And in this study, the researchers took a cohort of patients who had failed therapy for malignant melanoma, did the microbiota transplants, and after that, 30% of the patients then mounted an immune response. So the immune space is very, very exciting. The second space that is exciting is in the metabolic space. And here, and in this study, out of 15 patients who were enrolled in this with new onset type 1 diabetes, they essentially had cessation of the insulin resistance and the stoppages in the production of insulin following fecal microbiota transplantation. And over here in Parkinson's disease, patients improved dramatically after FMT. I would say that the science is very promising, but it's early days, and the future, at least for FMT, really lies in being able to better match donors to the right patients, because there's so much in that mix. Literally, the average microbiota profile is about 1 terabyte worth of data, thousands of different strains and different genuses. We need to understand more, a lot like blood in the early days, where we just knew when Napoleon's surgeons really transfused blood, that blood was blood. Today, we now know about the ABO blood types. We know how to fractionate blood into plasma, red cells, and so on and so forth. And we do see that this similar journey will happen, but for today, FMT is the most exciting application that we as doctors can put into clinical use almost immediately. And my two fellow speakers will talk about some of the specific strains and where the opportunities are. And as we understand more, we learn more, I'm sure that the next 10-15 years will be really, really very, very exciting for all of us. And for all of you out there who are in this space actively doing research, do feel free to give us a call and we can figure out how to work together to help further good research in gut microbiome science. And on this note, thank you very much. Ning, can I hand the time back to you?
Sure, thank you, Jeremy, very much for this very exciting talk. I think we will save the questions to the end. So next coming up, we will have Dr. Yan Tan, please.
Okay, yeah, thanks very much, Lynn. Let me share my.
Awesome.
Okay.
Can see it now.
All right. Okay, great. Yeah, thanks very much. And yeah, it's really glad to sort of like attend this event. And actually, we had a lot of like connections with Singapore before. We had a collaborator, Eric M, who started a lab in Singapore, and we also had actually a couple of interns in Singapore. So it's very exciting to join this event. And so my name is Yan Tan, and so I'm actually trained as a computational person, and mostly doing so at my very beginning of my PhD study, I'm doing like gene expression, and then gradually I started to do research in infectious disease and start to play with bugs. And at that time, so mostly we view microbiome as pathogens, right? And at the end of my sort of like a PhD training, we started to realize, okay, there's a lot of like beneficial microbes. We should really treat them as something would really play good in our health. So that's sort of like really intriguing to me. So after I finished my PhD study, I went back to China and joined the venture capital in China, and where I actually sort of like took this opportunity and dig into a lot of like companies in China. Most of them are data-driven or like gene sequence companies in China. And I figured out there's low sort of like a microbiome drug development company in China at that time, which is like almost like 3 years ago. So that's when I think this is really fantastic opportunity. And also there did be players in China. That's when we started to so like build this company, and we decided to adopt like a AI driven or like data driven approach in this sector. And so this might be like something everyone know or heard a lot before, especially I guess like audience for this event might be or should be very interested in the sort of like a relationship between microbes or like gut microbiome with human health and diseases. So I'll keep this sort of like introduction very brief. So there's like a couple of numbers I want to mention, which I was really surprised when I say those numbers, when I saw those numbers like a couple of years ago. So basically we now know there's like 10 kinds of number of cells or like gut microbiome cells than human cells in our body. So basically we carry more sort of like a gut microbes than actually human cells, which is like 10 times more. And also in terms of like a number of genes, right? It's almost like 100 times more. So that gives us a hint. There must be a lot of like functions microbes play in our body, and also give us a hint that there's a lot of like target or potentially for like a drug target. So we should really focus on that. And nowadays we know the gut microbiome is more like a forgotten organ. When we refer as an organ, it means it's really something commercial with human body, and we know it actually regulates our immune system, metabolic system, and the neural system, right? So as like Dr. Jeremy just mentioned, we know a lot of like diseases are associated with dysbiosis of human gut microbiome. That's how we started to think, okay, this is really, so I mean, before in the drug development industry, we focus more on the human genome. That's also when Broad Institute, where I trained as a PhD, started. And gradually, I think like even in our institute, we started to sort of like, we sort of like started to think, okay, we should like also care about the gut microbiome. And so Jeremy just like actually give us a very good introduction about FMT. in our opinion, if we think about like a modulation of gut microbiome as therapeutics, there are actually 4 routes, and at the very beginning, actually, it is FMT, and Jeremy also just introduced. And actually, the really interesting fact about FMT is if you take a look at the history of the therapeutics, it actually dated all the way back to ancient Chinese medicine. So at that time, it's called yellow soup, and ancient Chinese sort of like use that to treat GI symptoms like constipation or diarrhea, and that was like a couple of like 100 years ago, which is really fascinating. Like at that time, people already started to using the microbiome to treat diseases, even though we don't actually know what's effective sort of like mechanism for that. And nowadays, like FMT has already so sort of like proved in a lot of like clinical studies in a human trial. So I think it's already like widely used as a way to understand the microbiome and the human diseases. And also another good thing about FMT is the regulation is sort of like underway. I'll actually explain that later on. And there's a lot of like things we should think about FMT, as Dr. Jeremy just mentioned, like about like donor and donor recipient matching, etc. And another way is we call consortia. So basically it means like if we know exactly what are the sort of like microbes are playing treatment effect, we should actually identify them. We can isolate them and cultivate them and put them together as a recipe. And for that, so basically it's better things in terms of like the scalability, because it can actually leverage the fermentation industry as a whole. And the difficulty for that is like it's really it's still sort of like an open question, or at least like there's a lot of like different ways of identifying the effective like microbes, and also sort of like how to find the consortia. And one of the team in Xbiome is actually sort of like leveraging the data, the FMT data, publicly available, or we collected in China, and trying to use machine learning algorithms to do that. And so that's, I guess, some of the advancement in this field, and also sort of like there's a different approaches people are trying to find the best sort of like practice of doing that. And the third one we call metabolites, and actually I think we also include sort of like say epitopes or the peptides derived from the microbes. So basically this route we sort of like view as metabolites or the peptides derived from the microbes. So those could be like the MOA for the treatment effect of the bacteria. So if we actually know the MOA of the treatment effect, we can identify those molecules. We can use those molecules as a treatment. And also the interesting thing about this sort of like a route is it is I mean the way of finding those things are derived from the microbiome analysis. But the way of the drug development and also regulation fall into the traditional way of doing it, right? So it's like a small molecule or peptide drugs, and that clears the way of regulation and also drug development. And the only thing is basically how to find those metabolites, right? So that would be the sort of like both the potentials and challenges of this field. And the fourth one is synthetic bacteria. So I guess like the Dr. Jensen will address more later on. So basically, if we understand like the mechanism of the treatment effect of the bacteria, or for example the fish, we can actually synthetically or like biologically engineer those microbes and amplify their the effect, or make them deliver better. So that would be the 4 routes. And basically, I think that covers all the different routes people are doing in this field. And currently we are in a stage like we are accumulating a lot of the data and started to really find a way of doing other routes. And so as I mentioned about like the regulatory pathway, so I think that's when we're talking about like a therapeutics field. So regulations is always one of the most important things for us to care about. So just like 7 years ago, so which means like this field is actually really new. FDA announced the use of FMT is subject to the regulation by FDA. And so basically in 2014, FDA starts to require the stool materials for FMT must come from a donor who is known to the doctor or the recipient at the very beginning, and then start to change it as, okay, if the donor is from the stool bank. So I guess that also defines the industry standard at that time. And then on 2016, so there's sort of like a clear guidance, and this field is now designated as a live biotherapeutics. And on that guidance, there's a sort of like clear guidance about like the safety and the efficacy and CMC considerations for the IND application if we are in the space of the microbiome drug development. And here, actually, I get a table, and actually this is data I collected from another conference hosted in Singapore last year. Basically, compared the regulatory status from US, EU, Canada, Japan, and Korea. And actually, I'm very interested in like what's our current status in Singapore now. And basically, we can say like US has the best like or the most clear regulatory pathway, and EU and Canada start to follow, but they are on the way of draft more detailed like guidelines. And in Asia, we know like Japan and Korea is pretty active, and because like we are in China, we're actually now talking and communicating with the Chinese regulatory agency. And trying to see what they think about this field. And the good news is we know they are like the Chinese agencies are actually also developing, drafting the guidance. We think the new guidance will probably come out next year or the year after. And so this is a brief like introduction of Xbiome. We started on 2017. And as I mentioned before, we in China we adopt like a human first clinic study, sort of like a paradigm. We apply like IRB in the local hospital or the hospital collaborating with us, and we collecting the FMT treatment data. So basically, we collect the stool samples and other clinical or metadata from the patients before and after the treatment. And from there, we first evaluate whether FMT do have like a treatment effect on the diseases or indications we are interested in. And secondly, using this data, we are evaluating or trying to understand what are the microbes actually caused the treatment effect. So basically, we are trying to find out the causal effect. And at the very beginning, of course, like we collect like a public data, trying to figure out whether there's a strong association between the microbiome dysbiosis versus the diseases we are interested in. And currently we are around like 80 people, and mostly in Shenzhen, China. And we have a small small computational team in New York, US. We are actually very interested in collaborating with more research institute sort of like in Asia and also in US. And we collected a lot of data in China, and we are publishing a lot of them. And we're trying to share the sort of like data and also know-how here we've done in China. And this is the overview of the pipelines we are developing. I think like Jeremy just mentioned, like the immunology field is really exciting, and we are also believed in that. So we invested a lot, sort of like in the immunological field, and especially the immunotherapy. So we started like the first IIT trial in China using FMT treating patients who's not responding to the anti-PD-1 therapy. And also actually along the way, we already collected over sort of like I think over 4,000 samples already from the patients who's receiving the immunotherapies, and mostly of course like anti-PD-1. And we just submitted our first IND application I think last month to the US FDA, and the indication is acute GVHD. And next year we're actually thinking about just 3 new indications. One is C. difficile, and we know it works pretty well, and we want to also bring that to China. And also type 1 diabetes. I think Jeremy just mentioned one of the literature, and actually one of the authors is our collaborator on this. And also we are very interested in lupus now. And this is sort of like a way we are thinking about how to taking data from the hospital and using data-driven approaches, which means like bioinformatics pipelines, machine learning techniques, as well as integrated like knowledge base to figure out like what are the bacteria or combinations of bacteria or their metabolites might driving the treatment effect. And another part we call evidence-based, and basically after the computational approaches on the physical space we call, so we isolate the strains. So we are actually building a large library of strains, and currently we've already isolated more than 2,000 strains from samples, like for example the donor samples or the some of the patient samples. For example, we isolated like I think more than 70 species from the cancer patients who become complete responders after the anti-PD-1 treatment. And also we are building the Biotech animal model platform here in Shenzhen, which help us to sort of like scale up our capability of mechanism discovery of the microbiome functionalities, and also we think this would be a good animal model for us to test the hypothesis we've done or we've hypothesized from our computational model. And for the FMT drug, I just mentioned that we just like submitted the first IND application. I think that will be the first one in China. And we built our own CMC facility here in China, and we had a really close communication with the agency, and we really want to bring the sort of like the regulatory experiences from other countries to China and promote the development of that in China. So with that,
sorry.
Thank you.
Oh yeah, great.
Yes.
I was just about to say that we want to be cautious about time. Thank you.
Yeah. Okay. So this is last one. Thanks very much. So really good honor to attend this meeting. And as a CEO of a company, of course, we are hosting this one. We are hiring, and in US and China, and also we are really interested in the opportunities in Singapore too. Thanks very much.
Thank you so much, Yan. That's a very interesting overview of the field, and also a very important review of the regulatory path. And I know Xbiome is really a pioneer in trying to create a clear path for microbiome-based therapeutics in China. I think that's great work. Coming up next, we have John. Yeah, take it away, John.
Thanks a lot. Yeah, my name is Jonathan Kotula. I'm the head of science at Felix Biotechnology. We're a company in San Francisco, California. And I wanted to share a little bit about my background and my path to getting to be the head of science at Felix. So I consider myself an entrepreneurial scientist. The work that I've done has varied throughout the years, but it's always been focused on how do we get things to patients the fastest? So how do we take research discoveries that are really exciting that we all do at the bench and make them into therapeutics that help people? So I did a PhD training at Duke University in molecular genetics and microbiology. There, my PhD thesis was focused on catalytic RNA and using those as a delivery strategy for getting other catalytic oligonucleotides into cells. That work was really exciting and fun and was licensed to Roche, and that experience led me to really get involved in company creation and spinning work out of the lab. And so also while I was a grad student, I founded a small device company with the goal of preventing hospital-acquired infections. And that ties into something that I'm working on now. So we all know that antibiotic resistance is on the rise and is a really big problem. And a lot of the hospital-acquired infections, so infections that people get while they're in the hospital for something else, tend to be antibiotic resistant. And there need to be multiple solutions for solving that problem. And one that I worked on here was a way to increase hand washing among hospital workers to reduce the transmission of those infections. After my PhD, I joined Pam Silver's lab at Harvard Medical School and the Wyss Institute as a postdoc. What I wanted to work on is really learn all about synthetic biology, how to do genetic and genomic engineering in bacteria to generate engineered microbes to have an impact in a lot of different ways. When I joined her lab, I only vaguely knew of the microbiome. It wasn't something that I was, going there with a goal to generate tools for the microbiome. What I did work on when I joined the lab was a transcriptional memory device. And so this was a way to record events in bacterial genomes that they saw in different states and over different timeframes. This was the first demonstration of a bacterial engineering that functioned in a complex and competitive environment. And to show that it would do that, we thought we would test it in the microbiome. So we engineered these E. coli strains, first a lab strain and then a strain isolated from a mouse with the memory device, and then administered them to mice, and then showed that we could record events in the mouse gut and that the bacteria would remember the events that they saw and record those for months. Later work, we showed that it would, that memory lasted for months. We also use these tools to do some other interesting things that we think will be really interesting tools for how to probe the microbiome. And the one that I'm showing here is a cell division counter. So you can get a sense of how much growth is going on within the gut, which is something that is surprisingly difficult to measure. After leaving Dr. Silver's lab, I was the first scientist to join the company called SINLogic. SINLogic was based in Boston. It was one of the first companies in the US, at least, that was formed to do genomic engineering of microbes in order to use those microbes as living therapeutics. And so at SINLogic, we thought of the microbiome as a metabolic organ. So our liver is our metabolic organ, and there are a lot of people that are born with genetic diseases that are errors in metabolism. And so if you're born with one of these diseases, you can't metabolize certain food products. They can lead to the buildup of toxic metabolites and a lot of developmental problems for people that have these diseases. And our idea at SINLogic was that we could treat the combined metabolism of the microbiome as a metabolic organ, and one that we could engineer, and one that changes over with some frequency so that you could reintroduce or change the metabolism as you needed. So at SINLogic, I worked on a few different metabolism projects, including urea cycle disorder. So that's a disease where people that have urea cycle disorders cannot metabolize ammonia into urea. So you have toxic levels of ammonia built up in the blood, which is a serious problem. And so I engineered an organism, an E. coli nissel, to consume ammonia from the gut, prevent its absorption into the blood, and that living biotherapeutic, I led that project into clinical trials. It was in phase 1. It proved to be very safe, but we didn't hit the efficacy endpoints that were important for moving that drug forward. I also contributed a little to a PKU project, and that's phenylketonuria. So that's a metabolic disease where phenylalanine cannot be metabolized. And so that builds up in the blood, also leading to toxic effects. And so helped worked with a team that developed a bacteria that could consume phenylalanine. And that clinical trial is still ongoing, which is really exciting. I'm really hopeful that that's going to help a lot of people. After leading SINLogic, I joined the company Caribou Biosciences here in California. So Caribou is a company that was formed out of Berkeley to develop CRISPR-Cas9 into a useful tool to help people. And so when I joined Caribou, they wanted to start a microbial engineering group. So I came on as the associate director of microbial engineering and set up a team to develop Cas9-based tools to engineer gut microbes that we would isolate from people, not lab strains, and develop those into living therapeutics that would interact with the immune system within the gut. And so what I'm showing here is just a brief overview of the platform that we built, where we wanted to be able to identify, isolate, and characterize bacteria of interest from human patient samples. And so to do that, we developed a lot of tools for screening different microbes from fecal samples, culturing them on specific media so that we could narrow them down to the types of organisms that we wanted. We would do this in anaerobic chambers. We would do sequencing, both RNA and DNA, and really have a good characterization of the organisms that we wanted to work with. As you can imagine, if you do this work, that delivery of DNA into bacteria can be challenging, but is also essential if you want to do any genomic engineering. And so for the most part, all of us who work on bacteria and do synthetic biology or genomic engineering rely on tools like electroporation or conjugation or chemical competency to deliver DNA into those organisms. Excuse me. But we anticipated that by working with organisms that we were pulling directly out of patient samples, those tools or protocols may not exist. So we started developing a phage engineering platform where we would utilize phage transduction as our method of DNA delivery. And the idea was that we would we would engineer the phage to control what DNA was packaged into the phage and then control which organisms we delivered that DNA to. The tools we would be delivering, the DNA we would be delivering would be to express Cas9-based tools in order to edit the microbial genome. Converting the Cas9 tool into an effective genome editor is extremely difficult in microbes. And there's a lot of work that we did to get that to be successful. But what was really nice is that then enabled you to do rapid genomic engineering in a number of different organisms without having to develop unique tools for each organism. And then finally, when we would, the end result was we would engineer these organisms and then evaluate their interactions between the microbes and the immune system. And we would do this in mouse models. And the idea was that we would have interactions between the gut microbiome and the immune system to have impacts on immuno-oncology. And so there's a growing body of literature showing that the microbiome has an impact on whether patients are responders or not to checkpoint inhibitors. And we were developing ways to take advantage of that association and try to turn non-responders into responders. Earlier this year, this entire platform was sold to a different company called Novome, which is also in San Francisco, and is not being done at Caribou any longer. And so from there, I moved to Felix Biotechnology, where I'm the head of science. And so at Felix, we are a phage engineering company with the idea that we can develop novel living therapeutics to combat antibiotic-resistant infections. That's our first goal. But we can also engineer phage to deliver things, whether that's DNA to the microbes in your gut, or deliver precision subtraction of certain microbes from microbiomes. So if you can imagine that from the skin, if you want to subtract microbes that maybe cause acne or malodor, or if you think about that in the gut microbiome, if you want to subtract certain microbes that might have an association with cancer development, for example. But right now our focus is completely on antibiotic-resistant infections. And here I'm just showing something that I think we're all very familiar with, that antibiotics completely change the world. That because of antibiotics, we can now survive routine infections, we can have surgeries. They're really a miracle drug. But the problem is they're losing their effect through years of mismanagement from overuse, and just the fact of bacterial evolution. Antibiotics are losing their effectiveness, and that's going to be a huge problem for us in the future if we can't develop new solutions to antibiotic-resistant infections. And what I'm showing here is a woman, Paige, who has cystic fibrosis, which means it's a genetic condition that attacks the lungs, that leads to thick mucus buildup and infection, usually from Pseudomonas, that these patients will have their entire lives. And eventually the Pseudomonas infections will just be too much, and they will die from these infections. In May 2017, she was in the hospital with one of these infections that was resistant to all antibiotics, and her only chance at survival was a lung transplant, except she had heard about our technology of phage therapy. So we developed a phage cocktail for her, and which she received under an emergency access IND. The phage killed the bacteria, the Pseudomonas that were causing the infection, that improved her ability to breathe. And one of the unique attributes of our phage technology is any bacteria that evolves resistance to our phage necessarily becomes resensitized to antibiotics. So any bacteria that most of the bacteria we kill, any that survive that we don't kill, can now be retreated with antibiotics, which is, I think, a really exciting platform. So if you're not familiar with phage are viruses that kill exclusively bacteria, and they can be engineered to kill in different ways, and their tropism can be modified. We're really excited about them because they're very effective. They can kill both bacteria that are either sensitive or resistant to antibiotics. They're safe. They've been used for years, and there doesn't seem to be any toxic side effects of using a phage to a human, because they're so targeted to bacteria. They also can be so targeted that they don't kill beneficial bacteria as antibiotics do. So one of the problems with using antibiotics is complete destruction of the gut microbiome. Also a lot of toxic side effects. With phage, you don't have either. And then just in terms of building phage drugs, you only need a small dose, because as they infect, they generate more copies of themselves. They're simple to manufacture. You can do it in small bioreactors, and they can be engineered to have beneficial phenotypes like busting biofilms. So as I mentioned before, phage therapy has been around for a long time, well over 100 years. And so why haven't we heard of a general phage therapy yet? Part of the reason is that bacteria can rapidly evolve resistance to phage. There are a number of phage companies out there that are using cocktails that are trying to fight against this evolution by giving you a mixture of phages to try to combat that resistance. Our strategy, what makes us unique, is we're not trying to fight that evolution, but we are embracing it. So we are using phage that target, in this case, what I'm demonstrating here is a specific pump that leads to antibiotic resistance. So the phage recognize that pump, deliver their DNA, make copies of themselves, and then lyse and kill the bacteria. There are bacteria in the population that will have mutations to this pump, or they will delete this pump from their genome, and then thus survive the phage attack. And any that do that are now susceptible to antibiotic exposure, and so now will die from antibiotic delivery. And we can do this in cycles, and if necessary, and completely wipe out antibiotic-resistant infections. And so our platform, we think, is really advanced. We are taking advantage of machine learning and engineering. But first, what we do is we go phage hunting. We capture and identify, isolate new phage. We do that through phenotypic assays with wild bacteria, but also with knockout libraries, and that's what we're displaying on the top of this triangle circle diagram. When we find phage that are interesting to target the bacteria we want, we do a screening assay where we take hundreds of the phage that we isolate, and we can turn those into thousands upon thousands of phage through library generation through error-prone polymerases. And we can then screen all of those phages against our target bacteria and look for the phenotypes that we desire. And through sequencing, then we can put in, through sequencing, all the sequences of the phage, all the sequences of the bacteria, and use machine learning to find genetic elements that lead to the phenotypes that we want. We can then use that information to engineer the phage chassis that are easiest for us to work with. developing engineered phage that now have desirable attributes, whether that be alterations in tropism, the ability to bust biofilms, the ability to avoid CRISPR attacks from the native bacteria, whatever that may be. And then we're able to test those phage in people relatively quickly through emergency access INDs, which then, if they look to be effective and we're able to help people in that way, we can develop more controlled, larger scale clinical trials. And so today, that's what we're doing. We're working on phage that we've shown work well against Pseudomonas infections in cystic fibrosis patients. And we're working to engineer those and improve those phage and test them now in controlled, larger clinical trials. And we're at the point now where we're raising a fund. And so if you're interested in hearing more about us, please let me know. And if you're interested in joining the team, also please let me know. And that's everything here. So I'm happy to stop this and answer questions.
Thank you so much, John. That's a very exciting talk. Again, you are at the forefront of engineering biology applied to microbial therapeutics. So it's very great to see where the field has been advanced, latest, and also where the whole field is headed. Again, thank you so much to all the speakers. As we are running a little bit late, I guess we will have a more or less like compressed panel discussion. So maybe we will focus on some of the more general questions of general interest. So Yin Yang, please take the stage.
Oh, okay. Thank you for passing the baton. Actually, I'd like to thank all the speakers for an excellent talk and however brief it is you have of introducing the field that you guys are currently at. And quite some time that there has been some questions that has been already answered. So I'd like to start with a bit general high-level questions to further anticipate the questions from the audience. So I mean, each of three of you had different challenges and also different stages in clinical translation. Could you care to share a little bit more about the technical or business challenges that still remain despite all the technological advancements and the concessions that the governmental agencies are making in view of these translations? Are there any technical business challenges that are currently faced? Maybe Jeremy can, we'll just go in the order. Jeremy can start.
Sure. I mean, really young, as really Comrade Deng Xiaoping likes to say, we cross the river by feeling the stones. In this case, it's even harder than that. You have to build a bridge as you're crossing the river, because everything is really, the science is relatively young. Regulators are unfamiliar. Reimbursement mechanisms are non-existent. So, I mean, I am comforted that my boss used to tell me, my previous boss used to tell me, if it was easy, someone else would have done it way before you. So I think all of us come in with our eyes wide open that it's incredibly exciting. The potential for massive human impact is huge. But the science is challenging, and all the rest of the enabling ecosystem doesn't quite exist. And that's really why this makes the space so exciting, because we're not competitors or anything. We're all peers trying to grow this little mustard seed into something that is a lot bigger. And if gut microbiome science takes off, if synthetic biology takes off, there's more than enough space for everyone, and will have impacted and improved millions of lives around the world. So yes, and at least for us here in Southeast Asia, because I'll be very candid, Southeast Asia is very behind the curve, right? Beyond doing our own research, what we felt would help to jumpstart and catalyze would be to create the raw material, which is data, which are FMT specimens, and a general public, not just in Singapore, but across the region, who are aware that there's this thing called the gut microbiome, and if they contribute to the science, they can benefit from this science in the not so distant future.
Yep.
Yeah.
While we're on this topic, I'd like to extend this question to Jeremy and Yan. There has been a question in a Q&A which I feel needs to be addressed. So while on this FMT, I think the very question is, how do you then define healthy donor or the more or less effective donor FMT for C. diff has been recurring name and the usual suspect, but for the case of Yan, there's a whole list that this FMT is being applied to. So how do you actually then define the donor? Could you share a little bit before I address the question to John? Maybe Yan, could you?
Yeah, sure. Yeah. So I guess actually like here in so definitely it's very challenging problem I guess we are all facing in this field. And so here in Xbiome, we actually sort of like divide this question as two parts. So the first one is sort of like to find out the healthy donor. So which means like we do a lot of like tests, like blood tests and stool tests, and to sort of like sort out the pathogens from the stool, and that will be the healthy one. And so we actually, I mean, the interesting thing is like Dr. Jeremy just like mentioned, like you sort of like had this 2% rate of identify or finding like the healthy donor, and we also have like sort of like the same statistics. And as I know from Finch Therapeutics in US, and that's also their percentage. So I was actually wondering like why the numbers are so similar, right? Even though we are in different like places. So that would be the first one. So first one basically we sort out there's low pathogens in this donor. And the second one would be more sort of like challenging, which means to find out the right donor for the patient and also for the indication. And that's also two layers. Like the first layer is like you find a good like donor recipient like matching, which means you had a high success rate of engraft the microbiome translated into the patients. And the second layer is like for different indications, right? We need to find out different profile of microbiota. For example, like for the immunotherapy, we're actually looking for there's more microbes which can stimulate the immune system. And for some other diseases, we probably want to find like microbes which are deactivate the immune system, right? For some diseases. So that's like two layers. And here in Xbiome, we collecting samples and stools from donor first, and then we sequencing them and using our algorithms and also data collecting from public database and trying to develop like algorithms to do the matching. So both the donor recipient matching and also we call donor ranking. So basically rank the donor and see whether they are useful or they are compatible for different diseases.
Okay, so yours is primarily based on sequencing and plus other information. Jeremy, do you have anything to add on to that before I actually go to?
I actually think Yan gave a very comprehensive really response. I just want to emphasize that the so-called super donor for one disease state may not be a super donor in another disease state. So the complexity is I would suspect that as we learn more, we'll realize that the situation is even more complex than it already is.
So there's a possibility that the definition healthy donor may be different from the application down the street. I see. Okay, and I'll pass the question to John. What are the technical business challenges that are facing particularly phage therapy?
Yeah, that's a really great question. Some of the, if we start early in the process, getting clinical samples of bacteria from that we want to target, that is a challenge. Being able to have access to those and then kind of go phage hunting is what we call it. So being able to find a mixture of something that we hope will contain phage. So we know everything out there, everything that exists has phage in it, but will it have phage that are targeting the bacteria we want? So that is one technical challenge, but that's something that's really best solved by brute force, just testing as many things as possible until we find plaques that are doing what we want. Then of course, the challenge of engineering phage is substantial. We're working, I didn't mention in the talk, but we're working exclusively with lytic phage. So when you're working with lysogenic phage that incorporate into the genome, you can treat those like engineering bacteria, and there are a lot of tools for that, a lot of methods. When we're working with lytic phage, they don't incorporate into the genome, so you have to engineer in a very small window of when the phage deliver their DNA into the bacteria. Traditionally, that's how you would have to do it, but we're developing some novel ways to do engineering completely in vitro, and then reconstitute living phage without cells as well. And that's something that I'm really excited about, that we're hoping to take this field to the next level. And then after that, like once we have phage, it's manufacturing. So manufacturing living things in a quality way, in a GMP way, for clinical trials is extremely challenging. And most of the companies, at least in the US, are developing that capability in-house, which is quite expensive, but at this point is necessary. And so until there are really good, reliable contract manufacturing organizations that can do this for us, I think you're going to see that continue, where most people do it in-house, and that's a big challenge.
Right. Your talk earlier mentioned emergency access IND. How likely, I'm just moving the questions a little bit to regulatory turf now. How likely is it granted, and is it quite a common thing to grant or apply for?
Yeah, so I don't know how common it is across other diseases and other treatment options. With phage, there are most people who have been treated with phage, at least in the US, have been treated under this emergency access IND strategy. And the reason that it seems to, that the FDA seems to be okay with it with phage is throughout this, like I said, greater of people using phages, there have never been any toxic effects due to the phage. So they seem very, very safe, which is the FDA's #1 concern is like not hurting someone, right? So phages have this remarkable history of being safe and not causing toxicity in people. So if we're safe, and then on the other hand, these people are presenting with these really difficult, maybe impossible to treat antibiotic resistant infections with any other option, then that's the recipe for the emergency access IND.
I believe the panel discussion is actually coming to a close due to the time constraint. I would like to just round it off with one final question regarding each three of you's future perspective and where the field is going. Like, what is the next opportunity they may come or arise in respective company outlooks? Maybe we can start with Jeremy, and then Yan, and then Jonathan as well.
I think that the field will move as fast as there are resources, and I don't just mean money, I mean talent, laboratory talent, data science talent, and the regulatory permissiveness. For us in really AMiLI, while we are a platform, I had mentioned we are focused on some specific disease areas, and we have chosen inflammatory bowel disease, largely because two of my co-founders, they are subspecialists clinically in this area. So no issues with patients, with no issues with clinical trials and really so on. And the other area that we're looking at is in the space of liver disease. That's because here in Asia, liver cancer is far more common than it is in Western countries. And so these are the two areas that we are looking at, but we again see AMiLI as a platform. So we do work with multiple institutions across Southeast Asia on everything from really addiction science to autism and so on and so forth.
Yeah. Okay, great. Yeah.
Yeah. So I think like this field will definitely like advance much faster. And so we see that trend already. And I guess like the advancement of this field will depend on much more tools. And when I refer to tools, I mean both like computational tools and also screening tools, like for example some high throughput methods to screen the functions of the strains, and also synthetic biology type of tools. And I guess like a lot of the things we saw, a lot of the data we saw give us a strong association between the microbes and the the human diseases and health. And the next step would be like how to actually find the causality and also further illustrate the MOA of the treatment effect. And that's also important because like the better we illustrate the MOA, the easier we communicate with the regulatory agency and also sort of like help them to make policies for this field. And that's very important because for therapeutics, right? So the agency is one of the most important sort of like players in this industry. And for Xbiome, so we are focusing on various like spectrum of diseases. And one of the most important one would be the immune related diseases, because we saw gut microbiome as a very important modulators of our immune system. So that's one of the sort of like area we are betting a lot about.
Lovely. John?
Yeah, I think the field is ready to just grow exponentially in the US, and people are just waiting for some exciting clinical victories, so some successes. There are a lot of really exciting companies out there, from people who are doing FMTs and consortia like Finch and Ceres and Vedanta, and then companies that are doing now microbial engineering, like SynLogic, like Novome, and then companies like ours that are starting to do phage engineering. It's just, I think we're right on the edge of just this growing extremely fast, and there are a few active clinical trials, and as we start to see successes from that, I think there's going to be a lot of excitement from the investment side, because there's already seems to be a lot of excitement just from people who are like who are learning about the microbiome and are really interested in treatments that would come from that.
I think the potential in the future for microbes as therapeutics is quite bright, and especially with quite a academics as well as companies, and the products have or is entering the clinical phase trials and very close to being available in clinics and actually really addressing some of the unmet medical needs. I can only look forward to what it's going to shape out to be in next coming years. With that, I'd like to actually pass it back to Ziying.
Yeah. Oh, sorry about the background. Yes. Okay. Yes. Yeah, thank you, Dr. Jeremy Lim, Dr. Yan Tan, and Dr. Jonathan Kotula for your insightful presentations, and Dr. Huang for your great moderation. And of course, thank you to all our attendees for your time and questions. It's a pity that we run out of time, and hopefully the discussion can continue and grow offline. We hope this webinar sparks more interest in the innovative science and technology that microbes hold, and we hope you come away from this session with more knowledge and insights. Do keep an eye out for a post-event email with the recording to the sessions and ways to connect with our speakers. So until we meet again, bye-bye.
Thank you so much, everyone. Bye-bye.
Yeah,
Thank
you very much.
Bye-bye. Have a good week.